Hello everyone. Today we will discuss about hemoglobin. Hemoglobin is the red coloring matter of the blood. It is a conjugated protein and a chromoprotein. And it contains heme as the prosthetic group. Heme as the prosthetic group and globin as the protein part. It's a conjugated protein. It contains heme as the prosthetic group and globin as the protein part. So next we will see the structure of hemoglobin. The structure can be discussed under two headings. Structure of heme, that is the prosthetic group, and structure of globin, that is the protein part. First we will see heme. Coming to heme, heme is an iron porphyrin compound. Iron porphyrin compound. That means it contains iron and porphyrin. So what is the porphyrin? Porphyrins are complex compounds with a tetrapyrrole structure. This is the pyrrole ring. This is pyrrole ring. So porphyrins are complex compounds with a tetrapyrrole structure. Tetrapyrrole means 4 pyrrole rings. So 4 such pyrroles, that is 1 to 4, 1, 2, 3, 4. These 4 pyrrole structure combined through CH bridges. These bridges are called as methene or methylidine bridges. Methene or methylidine bridges. So porphyrins are compounds containing tetrapyrrole structure. So 4 such pyrroles, that means tetrapyrrole, that is 1 to 4 combined through this methene or methylidine bridges to form a porphyrin nucleus. This is the entire structure of hemoglobin. This is heme and it is connected to a globin that forms hemoglobin. Here these are pyrrole rings. 1, 2, 3, 4 are pyrrole rings. Here these are alpha, beta, gamma, delta. This represent these methene or methylene rings. Here M means methyl group. M is methyl group. V means this vinyl group. P means propionic acid. Here 2 propionic acid. Here MV. MV. Here also M. Here also M. That is methyl group. Then methylene. We can represent these methylidine bridges as alpha, beta, gamma and delta. This is the structure of heme. Then coming to the structure, the 2 hydrogen atoms of NH groups of the 2 hydrogen atoms of NH group of 2 and 4 pyrrole rings. The hydrogen atom, here 1 hydrogen atom, here 1 hydrogen atom of 2 and 4 pyrrole rings are replaced by the ferrous ion. Here Fe++, ferrous ion, which occupy the center of the compound ring structure. This ferrous ion is occupied in the center of the compound structure and it establishes linkages with all the nitrogen atoms of the pyrrole rings. The ferrous ion is also linked to the nitrogen of the imidazole ring of this histidine of the polypeptide chain. Ferrous ion is also linked to the nitrogen of imidazole ring of this histidine of the polypeptide chain, that is heme linked group. Then the propionic acid, here propionic acid group, COH group. Propionic acid COH group. Propionic acid is CH2 CH2 COH. So the propionic acid COH group of this 3 and 4 pyrroles are also linked to the arginine and lysine of the polypeptide chain. The propionic acid COH group of the 3 and 4 pyrrole rings are also linked through arginine and lysine to the polypeptide chain. If this is the central ion, that is Fe2+. If the central ion is oxidized, it gets converted to Fe3+ state. If it is oxidized, it is converted to Fe3+ state. Then it will carry a positive charge. This positive charge is balanced by taking an OH group from the medium. It is also balanced by other ions like Cl- or SO4-, chloride or sulfate if available. That is the structure of heme. Coming to globin, it is the protein part of hemoglobin. It is composed of 4 polypeptide chains. 4 polypeptide chains. That is 2 identical alpha chains and 2 identical beta chains. So, 4 polypeptide chains in this protein part, that is globin. These 2 identical alpha chains and 2 identical beta chains arranged as a tetrahedron. These are arranged as a tetrahedron. Each polypeptide chain contains a heme. Each polypeptide chain contains a heme. That is in the heme pocket. Heme pocket. Thus, one hemoglobin molecule contains 4 heme units. Each polypeptide chain contains a heme that is in the heme pocket. So, one hemoglobin molecule contains 4 heme units. So what is this heme pocket? We will see the formation of heme pockets. Hemoglobin molecule and its subunits contain mostly hydrophobic amino acids internally. Hemoglobin contain hydrophobic amino acids internally and hydrophilic amino acids on their surfaces. Hydrophobic internally and hydrophilic on the surfaces. So, the hemoglobin molecule is waxy inside. Waxy inside. Because internally there is hydrophobic amino acid. So, it is waxy inside and soapy outside. It is soapy outside. So, this making it soluble in water. It is soluble in water and impermeable to water. Impermeable to water. So, it is soluble in water but impermeable to water. Then, each subunit contains one heme moiety hidden within a waxy pocket. So, each subunit contains one heme moiety that is hidden in the waxy pocket, that is inside, that is hydrophobic amino acids. The heme pockets of alpha units are of size just adequate for the entry of oxygen. The heme pockets of alpha units are of size just adequate for the entry of this oxygen molecule. But the entry of oxygen into the heme pockets of beta subunit is blocked by a valence residue. So, this oxygen, the heme pockets of alpha units are of size just adequate for the entry of oxygen molecule. But the entry of oxygen into the heme pockets of beta subunit is blocked by valence residue. So, that is about the structure of hemoglobin. Next, we will see the various types of normal hemoglobin. This is about the structure. Next, we will see varieties of normal human hemoglobin. So normal hemoglobin is of several types. It contains 4 subunits. Contains 4 subunits made up of various combinations of, if possibly, 5 different or related polypeptide chains. It contains 4 subunits that are made up of various combinations of 5 different or related polypeptide chains. These are designated as alpha, beta, gamma, delta and epsilon. Most normal human hemoglobin contains 2 alpha chains plus 2 other chains. The other chains may be beta, gamma, delta or epsilon. So, most normal human hemoglobin contains 2 alpha chains plus 2 other chains. The other chains may be beta, gamma, delta and epsilon depending on the type of hemoglobin. So, first one is HbA1 or HbA. HbA1 or HbA. This is the normal adult hemoglobin. Normal adult hemoglobin. This is the adult hemoglobin. It is called as HbA. It consists of 2 alpha and 2 beta. 2 alpha and 2 beta chains. So we can designate it as alpha A2 beta A2 or simply as alpha 2 beta 2. Alpha 2 beta 2 means 2 alpha, it contains 2 alpha chains and 2 beta chains. It is the normal adult hemoglobin. So it is about 90 to 95% of hemoglobin of normal adult. 90 to 95% of hemoglobin of normal adult is this HbA1. So next we will see the difference between alpha and beta chains of adult normal hemoglobin, that is hemoglobin A or A1. Difference between alpha and beta chains. So it is alpha to beta 2. It contains 2 alpha and 2 beta chains. So first difference is here the total amino acid is 141 and in beta chain it is 146. Coming to C-terminal amino acid, it is arginine. In beta chain it is histidine. Coming to N-terminal amino acid, it is valine, leucine. Here the N-terminal amino acid will be valine, histidine, leucine. Valine, histidine, leucine. Then coming to alpha helices, 7, 8. Coming to the heme pocket, in alpha subunit it is adequate for entry of 1 molecule of oxygen. But here in beta, entry of oxygen in heme pocket is blocked by a valine residues. These are the difference between alpha and beta chains of adult normal hemoglobin, that is HbA. So total amino acid here is 141, here it is 146. The C-terminal amino acid in alpha subunit is arginine, here it is histidine. N-terminal amino acid is valine, leucine in alpha chain, valine, histidine, leucine in beta chain. Coming to alpha helices, it is 7, here it is 8. Then about heme pocket, in alpha it is adequate for the entry of 1 molecule of oxygen. In beta subunit, entry of oxygen in heme pocket is blocked by a valine residue. That is about HbA1, that is the adult hemoglobin. That is about 90 to 95% of hemoglobin of adult type. Then next one is HbF. So F, as the name indicates, it is the human fetal hemoglobin. Human fetal hemoglobin. It can be designated as HbF and it is alpha 2, gamma 2. Adult HbA is alpha 2, beta 2. Here fetal hemoglobin is alpha 2, gamma 2. And this HbF differs from this HbA1 you will see the difference between HbA and HbF. HbA and HbF. So what are the differences? Here the polypeptide chain is alpha 2 beta 2. That is the first difference. Here it is alpha 2 gamma 2. Then in case of HbA, at pH 8.9, HbA moves ahead of this HbF in electrophoresis. So in electrophoresis, HbA moves ahead of the HbF. So HbF moves behind HbA in electrophoresis. Then in the case of HbA, affinity towards oxygen is less. Here affinity to oxygen is more. Coming to concentration, about HbA at birth it is about HbA is about 85%. At birth it is 85%. This HbF is 15%. Then this HbF disappears by end of first year. HbF disappears by the end of first year. So persistence of this HbF after 1 year is pathological. So it is disappeared by the end of first year. Persistence of HbF after 1 year is pathological. In this HbA in adult, concentration is about 90 to 95%. That is the difference between HbA and HbF. HbF is the fetal hemoglobin alpha 2 gamma 2. Next one is HbA2. It is a minor component of normal adult hemoglobin. Major component is HbA1 or HbA. This is the minor component of adult hemoglobin. It is usually about 25%. Electrophoretically, it is a slowly migrating fraction. Slowly migrating fraction. It contains 2 alpha chains and 2 delta chains. So it is alpha 2 delta 2. 2 alpha and 2 delta chains. That is HbA2. It is a minor adult hemoglobin. It is about 25%. It is in electrophoresis. It is slowly migrating factor. It can be represented as alpha 2 delta 2. It contains 2 alpha and 2 delta chains. Next one is embryonic hemoglobin. Embryonic hemoglobin. This is found in first 3 months of intrauterine life of the baby. First 3 months of intrauterine life of the baby. It is found in first 3 months of intrauterine life of the baby. It contains 2 alpha chains and 2 epsilon chains. So it is alpha 2 epsilon 2. 2 alpha chains and 2 epsilon chains. Embryonic hemoglobins occur in human erythrocyte in the first 3 months of embryonic life. It occurs in human erythrocytes in first 3 months of embryonic life. That is embryonic hemoglobin. Next one is HbA3. HbA3 and electrophoretically fast fraction. It is fast fraction. It is about 3 to 10% of total hemoglobin. This is designated as A3 and it is an altered form of HbA. It is an altered form of HbA. It is found chiefly in old red cells. So electrophoretically it is a fast fraction. It is about 3 to 10% of total hemoglobin. It is an altered form of HbA and it is chiefly found in old red cells. That is HbA3. Next one is HbA1c or glycosylated hemoglobin. HbA1c. In addition to HbA1, HbA1 means that is the major form of normal adult hemoglobin. A minor glycosylated form is also found in adult red blood cells. Minor glycosylated form. It is also found in adult red blood cells. That is called HbA1c. It is otherwise called as glycosylated hemoglobin. It is about 3 to 5% of the total hemoglobin. In patients with the diabetes mellitus, it may be increased to 6 to 15% of the total hemoglobin. In patients with diabetes mellitus, it may be increased to 6 to 15% of the total hemoglobin. Then amino acid sequence of this HbA1c is similar to that of HbA1. Amino acid sequence is similar. The chemical difference is attachment of 1 amino 1 deoxyfructose 1 amino 1 deoxyfructose to amino terminal valine of beta chain of HbA1c. HbA1. So amino acid sequence is same in that of HbA1. The chemical difference is the attachment of 1 amino 1 deoxyfructose to the amino terminal valine of beta chain of HbA1. That is the chemical difference. This addition of this sugar, that is 1 amino 1 deoxyfructose moiety to the valine occurs non-enzymatically, either by addition of glucose directly to the protein or by the formation of glucose 6-phosphate. Then which is subsequently dephosphorylated to form the HbA1c. So this addition of sugar moiety to the valine occurs non-enzymatically, either by addition of glucose directly to the protein or by the formation of glucose 6-phosphate of an adult hemoglobin, that is HbA1. This is subsequently dephosphorylated to form HbA1c. Then the level of HbA1c is an index of the levels of blood sugar for a period of several weeks. The level of HbA1c is an index of the levels of blood sugar for a period of several weeks prior to this time of sampling. Once the RB cells get glycosylated, it remains free for the lifespan of the RB cells. Its measurement is a more reliable indicator of the adequacy of control of diabetic state as compared to occasional measurement of blood and urine glucose. So, this HbA1c measurement is a more reliable indicator. More reliable indicator. Its measurement is a more reliable indicator of the adequacy of control of diabetic state as compared to occasional measurement of this blood and urine glucose. So that is about HbA1c. So in total, soon after the fetus begins to develop, there is a rapid production of alpha and epsilon chains. Then the alpha chains persist, whereas epsilon disappear. Then a new polypeptide chain, that is gamma chain appear. But the alpha chain persists. Then gamma chain appear. During the later stage of this fetal growth and after birth, beta chains increases in quantity. During the later stage of fetal growth and after birth, beta chains increases in quantity as the gamma chains decreases correspondingly. So coming to the properties of this hemoglobin, it's a crystalline protein. Each species has its own crystalline form. Each species has its own crystalline form which is related to variations in amino acids of the globin part. Coming to molecular weight, approximately 65,000 as determined by the osmotic pressure and ultracentrifugation measurements. Coming to shape, the overall shape resembles a spheroid. So, this is about the structure and varieties of normal hemoglobin. So, this is today's topic. Thank you for watching.